So let's quickly look at the various ways that antibiotics can actually work to basically kill bacteria or inhibit their actions. All right. And first of all, before we go into this lecture, at least you should know what an antibiotic is. All right. Antibiotic is any drug or agent that will act against a bacteria. Okay. So acting not really okay. Yeah, bacteria. Okay, bacteria. This one is specific to bacteria. All right. So acting against bacteria, they basically have different points where they act. Do you understand? So these points where they act is what is known as what mechanism of action. Okay, so be picking each of these points, to be describing it and be talking about the relevant examples to it. All right. So the full question, as it was asked in an exam, was enumerate the different mechanisms of actions of what antibiotics with relevant examples. Right. So you can see what antibiotics are different areas where they could work. Inhibition of nucleic acid synthesis. Don't worry, we have the full lectures here. All right. I just want to just refresh your memory. They could inhibit nucleic acid synthesis. They could inhibit protein synthesis. They could inhibit. Okay, that's what the 50S RNA. They could also inhibit protein synthesis of 30S RNA. They could inhibit cell wall synthesis. All right. They could alter cell membrane and synthesis. All right. They could inhibit folate synthesis, inhibit DNA dependent RNA polymerase. All right. They have different mechanisms of action, which we'll be looking at. So the first one is what? Inhibition of cell wall synthesis. And I've already made this image dirty, so I would like to just use the next one. Where's cell wall here? It is cell wall synthesis. All right. So it will basically what? Stop the bacteria from forming a cell wall. Now, if there's no cell wall, the bacteria will not be resistant, okay, to damage, okay. That's just literally what it means. So inhibition of cell wall is like this. All lactam drugs are what selective inhibitors of bacterial cell wall synthesis, and therefore they are active against what growing bacteria. So basically what inhibits a growing bacteria, they'll inhibit it from growing to full potential, because before a bacteria can grow to full potential, it has to have the cell wall. Okay. Now this is the initial step in drug action, and it consists of binding of the drug to the cell receptors. That's the penicillin binding proteins. Okay. So now the drug acts by inhibiting the transpeptidase enzyme. Now, inhibition of the transpeptidase enzyme by penicillin and cephalosporin may be due to a structural similarity of these drugs to the terminal D-alanine of the pentapeptide. Okay, so now you can see examples of drugs that can inhibit the formation of bacterial cell wall. We have the beta lactams, we have the glycopeptides, we have the phosphomycin, we have the bacitracin, you have the what alaphosphalline. Okay, and moving to the next one, we have inhibition of protein synthesis. Let's look at it on this image. All right, you can see here inhibition of protein synthesis, and we have the 30S and the 50S inhibitors. Okay, so inhibition of protein synthesis is like this. Bacteria have what? 70S what? Ribosomes, right? They have 70S ribosomes, whereas the mammalian cells have what? 80S ribosomes. Okay, so now the subunits of each type of ribosomes are what? Sufficiently different to explain the reason for what? Selective toxicity. Okay, so you could see that what the aminoglycosides and tetracyclines can inhibit the 30S subunit ribosomes. So now if they are inhibiting the ribosomes, what are the primary functions of ribosomes? Formation of proteins. And proteins are vital components of life. Okay. So if the aminoglycosides and tetracyclines are inhibiting the 30S subunit ribosomes, then you know that what? Formation of proteins is basically what? Obstructed. Then the chloramphenicol and macrolides can inhibit what? The 50S subunit of what? ribosomes. So looking at here, you can see them here. This is how protein synthesis is basically inhibited. Okay, and these are the drugs that can inhibit protein synthesis, right? We have the tetracyclines, aminoglycosides, we have the macrolides, we have the lincosamides, we have the word amphinicols, we have the pleuromutilins, and we have the oxazolidinones. Okay, so moving to the next one is what? Inhibition of nucleic acid. That's inhibition of what? Formation of nucleic acid. Now, do you guys hear? Inhibition of nucleic acid. Yeah. This is DNA gyrase inhibitors. Kind of. Let's read it first. Yeah, DNA gyrase. Okay. So DNA gyrase catalyzes what? Negative superhelical twist in circular chromosomes and plasmid DNA. You know, before you have a DNA, all those two helical strands need to twist. All right. So basically, if you inhibit the twist of those strands, DNA will not be formed. Okay. So that's just the basis. Then another enzyme known as the topoisomerase 4 resolves interlinked daughter DNA molecule to allow their segregation into what? The daughter cells, right? So quinolones what? Inhibits the above two enzymes. You know these two enzymes, the topoisomerase and the DNA gyrase, they are basically what? Facilitating or promoting the formation of what? DNA strands. Okay? But if you apply quinolones now, quinolones will inhibit the above two enzymes, thereby promoting the cleavage of DNA. Alright? Promoting the breakdown of DNA. Okay? So that's it. And we're moving to the next one. Then there's inhibition of DNA dependent what? Polymerase. I like using this image. All right, so you can see inhibition of DNA dependent what? Polymerase. This one basically will inhibit the mRNA. Okay, so let's see how it acts. So an example of drug that will inhibit DNA dependent polymerase is what? Rifampin. Rifampin is an inhibitor of RNA synthesis by inhibiting the enzyme DNA dependent what? RNA polymerase. Okay. Then also inhibition of metabolic pathway. Inhibition of metabolic pathway. Well, let's read about it. All right. So inhibition of metabolic pathways. You have the p-amino benzoic acid. That's a PABA. Is involved in the synthesis of what? Folic acid, which is an important precursor in the synthesis of what? Nucleic acid. All right. So sulfonamides are structural analogs of what? That's p-amino benzoic acid. So they'll act like p-amino benzoic acid, but they'll basically inhibit the process. Okay. So now they can inhibit what? The dihydropterate. All right. So this was actually what? Folate synthesis. Okay. Inhibition of folate synthesis. Okay. The next one, there's inhibition of cell membrane. So inhibition of cell membrane, we have drugs like what? Polymyxin, colistin, and the daptomycin. Right? So these are what? The various mechanisms of action of what? Different antibiotics. All right? Different mechanism of actions of different antibiotics. That's how I'll just name it. All right? Because we spoke about different mechanisms, we also spoke about different examples of antibiotics. So that's it, and bye for now.